Have you ever really stopped to think about how something as huge as a redwood tree, or, even you, started out as just one single tiny cell? It's one of the biggest questions in biology. So today, we're going to unpack that incredible journey from one cell to trillions.
I mean, just think about that for a second. The trillions of cells that are you right now, the ones that let you grow, the ones that heal a cut, they all came from that one original cell. It's this one process of division that connects you, me, and every single living thing on the planet. So yeah, that's the big question, right? How does one cell manage to multiply into the trillions, making sure every single new cell gets a perfect copy of the instructions? And how does it also create the super specialized cells we need for reproduction? Well, the answer lies in two just beautifully elegant processes. Okay, so first things first. Before a cell can even think about dividing, it has to get ready. This isn't just some random event. It's a super regulated, carefully timed sequence we call the cell cycle. You can think of it as all the crucial prep work before the main event. What might surprise you is that the actual division part, what scientists call the M phase, is actually the shortest part of the cell's life. Like maybe just an hour. Over 95% of its time is spent in this preparatory stage called interphase. And trust me, it is a period of intense activity. So this interphase, it's got three parts. In G1, the cell basically just grows and does its normal cell stuff. Then comes the most critical part, the S phase. S stands for synthesis. And this is where it meticulously copies its entire set of DNA. And finally, in G2, it makes the last few proteins it needs and runs a final systems check before the big divide. And really, that's the whole point of the S phase. It's making a perfect, identical copy of all its DNA. This duplication is absolutely essential to make sure that both of the new daughter cells get a complete and perfect set of genetic blueprints. Now stick with me, because this is a really key point that can be a little confusing. After the S phase, the amount of DNA in the cell has literally doubled, but the number of chromosomes is technically still the same. How is that possible? Well, it's because the original strand and its new copy, we call them sister chromatids, stay glued together, forming that classic X shape that we all think of when we picture a chromosome. All right, so the cell's prepped, its DNA is copied and ready to go. Now it's time for mitosis. This is the process for creating two genetically identical daughter cells. It's the engine of growth and repair in our bodies. A great way to think about mitosis is like a carefully choreographed four-act play. Now, it's really one continuous process, but breaking it down like this really helps us appreciate the incredible precision that's at work in every single stage. So in Act 1, prophase, all that long, stringy DNA, the chromatin, starts to condense. Just imagine a bunch of tangled yarn being neatly coiled up into those distinct chromosomes. At the exact same time, the nucleus wall dissolves, basically clearing the stage for all the division machinery to get to work. Act 2 is metaphase. This is where all those X-shaped chromosomes get lined up perfectly right down the middle of the cell. These tiny protein threads called spindle fibers attach to each one, creating this kind of molecular tug-of-war that holds them in perfect alignment. The cell is now at the supercritical checkpoint. All the chromosomes are lined up, they're under tension, just poised for what is easily the most dramatic moment of the entire cycle. This is the climax of the play. And boom, this is anaphase. The connections holding those sister chromatids together just snap. In this swift, coordinated move, the chromatids, which are now considered their own individual chromosomes, get yanked apart to opposite ends of the cell. It's incredible to watch. And finally, in telophase, the chromosomes arrive at their destinations and start to unwind. Two brand new nuclei form around the genetic material. Then, in one last step called cytokinesis, the cell itself actually pinches in the middle, splitting the rest of its contents to create two brand new genetically identical cells. Mission accomplished. Okay, so mitosis builds and repairs the body with perfect copies. But what about reproduction? I mean, for that, you need a completely different process, one that's designed not to copy, but to create genetic variety. And that process is called meiosis. Right, so the crucial difference is this. If mitosis is a copy machine, meiosis is more like a genetic shuffling machine. Its whole goal is to produce sperm and egg cells, we call them gametes, that have only half the number of chromosomes and are all genetically unique. So how does meiosis pull this off? Well, it has a few key tricks. First, it involves 2 full rounds of division, not just one. Second, what we call homologous chromosomes. That's the one you got from your mom and the one from your dad for each pair. They actually pair up. And third, while they're paired up, they literally swap pieces of DNA. This is an event called crossing over. The first phase, prophase 1, is where the real magic of genetic shuffling happens. Those homologous chromosomes find each other and pair up so tightly they can physically exchange little segments. This process, crossing over, creates brand new combinations of genes on each chromosome. It is the primary reason why siblings aren't identical. And here's the other huge difference in the divisions. In that first round, during anaphase 1, it's the homologous pairs that get separated, not the sister chromatids like in mitosis. Then, the second division, meiosis 2, actually looks a whole lot like mitosis. This time, the sister chromatids finally get pulled apart.
So what's the final result? Not 2 identical cells, no way. You get 4 genetically unique cells, and each one has just half the number of chromosomes as the original parent cell, which makes them perfectly ready for fertilization.
So there we have it. These 2 fundamental processes of division, each one perfectly suited for its specific job. Let's just put them side by side for a final comparison. This chart really sums it all up, doesn't it? Mitosis is all about growth and repair. It does 1 division to make 2 identical cells. Meiosis, on the other hand, is for making gametes for reproduction. It does 2 divisions to make 4 unique cells, all thanks to that amazing trick called crossing over. That's really the core takeaway here. So mitosis ensures stability, faithfully copying our genetic code over and over for a lifetime. But meiosis ensures diversity, constantly shuffling the deck and creating new gene combinations with every single generation. This incredible balance between copying and creating, it's not just happening inside ourselves. It is the very engine that drives the evolution of all life on Earth.